decía, muchas gracias por estar aquí. Esta parte de este evento es el primer paso que damos para el inicio de actividades de colaboración que se tienen planteadas, que se contemplan para vincular a la FES Iztacala con la Universidad de Turín. En este caso, vamos a tener una conferencia dictada por la doctora Ludovica y se piensa con el apoyo de la División de Investigación, con el apoyo de la Secretaría de Relaciones Institucionales y, por supuesto, de la Dirección de la FES Iztacala, que llevemos este convenio a un beneficio académico mutuo donde podamos tener intercambios de estudiantes, de profesores y una colaboración exitosa en investigación. Todo este trabajo tiene de antecedente todo el proyecto que ya hemos seguido durante muchos años con el Royal Kew Garden y que ha sido a través de la doctora Tiziana Ulián, que también nos acompaña en esta ocasión. Y bueno, pues es eso. Yo les hice una invitación muy cordial y atenta a todos, convencido de que es una oportunidad de tener el contacto con el trabajo que se hace en otros institutos, en otras universidades, y como un preámbulo para poder extender nuestra visión del trabajo. Bueno, voy a hacer una pequeña presentación de la doctora. Es que yo veo un micrófono y quiero cantar, pero a ver. Vamos a tener la presentación, la conferencia de la doctora Ludovica Oddi, que lleva por título Evaluación del secuestro de carbono en diferentes tipos de vegetación en los Alpes, cuáles son las tendencias y cuáles son los retos de este trabajo. Muy brevemente les comento que la doctora Ludovica Oddi es una bióloga ambiental especializada en ecología vegetal y procesos ecosistémicos. Ella actualmente trabaja como investigadora y profesora en el Departamento de Ciencias de la Vida y Biología de Sistemas de la Universidad de Turín en Italia. Bueno, hizo su doctorado en el periodo de 2015 al 2018 y se centró su estudio en la heterogeneidad ambiental como impulsora de la biodiversidad estructural y funcional de los sistemas alpinos, que es pues donde está cercano su centro de trabajo. Posteriormente, la doctora Ludovica Oddi amplió su enfoque y su materia de estudio, su línea de investigación, para entender cuál es la heterogeneidad, la diversidad y el funcionamiento de diferentes tipos ecosistémicos, con el objetivo principal de abordar cuestiones ambientales y vincularlas con decisiones de política científica y política ambiental. La doctora Ludovica Oddi es una practicante constante de la ciencia del uso de la investigación básica como fuente de conocimiento, y también utiliza los medios y los recursos y las herramientas modernas para el análisis de sus datos. Ella está convencida de que, independientemente del enfoque que haga de su trabajo, las respuestas, como ella lo dice, las respuestas siempre están en la biología. Les dejo a Ludovica Oddi. Muchas gracias.
Gracias, Cesar, for this nice introduction, and thanks to all of you for being here. So, as Cesar already said, today I'm going to talk to you about the carbon sequestration in the Alps in different types of vegetation, and we'll see together some methods and some challenges that the ecologist has to cope with in this kind of ecosystems. Has any one of you been in the Alps? No? Who knows? So everything is new, and this is nice. So just a brief outline of what I will talk about today. It's a short introduction about me, and then a brief overview of the Alps so you can know better these ecosystems, and then some informations about carbon cycle, and then I'm going to present you 3 case studies that I followed in the Alps, and then briefly new projects and perspective. So I need to go back. Okay, perfect. So I studied natural sciences and I got my bachelor's degree in 2012, and my dream was to become a marine biologist. So as you can see, okay, as you can see, something went really wrong because I ended up studying like Arctic and alpine ecosystems. This is because, am I changing slides or you are changing slides? No, just to know if I have to ask or if I can do with this. Okay, now it's fine. So I got my master's degree in environmental biology in 2015 at the University of Torino, and then the PhD in 2018 in biological sciences, and I did half of my PhD in Torino and the other half in Scotland at the James Hutton Institute of Aberdeen. And then I've been postdoc for 4 years, and now I'm a researcher and lecturer in plant ecology and conservation. So I'm actually from Rome, but I moved to Torino, as you can see here, that is very, very close to the Alps, and that's why I didn't become a marine biologist. As you can see, this is Torino, beautiful city that is very, very close to the western Italian Alps. So the alpine biogeographical regions, this is the distribution, as you can see, the purple one. This is the distribution of this alpine biogeographical region. As you can see here, we have it in Italy, but we have also a bit of this region on the Apennines, and then in other mountain systems of Europe, as Pyrenees here, and so on. In the Italian Alps can be like divided in 3 main areas that are the Western Alps, Central Alps, and Eastern Alps. All these areas present very different characteristics, from like soil properties, climate, and vegetation as well as a consequence. So today we will focus on the Western Alps because are the one that I studied more, that are closer to Torino. And okay, these pictures just to show you some of the most important peaks that we have on the Western Alps. The Mont Blanc is the highest mountain in the Alps. That is also the highest mountain in Italy. And then the Cervino, that is also is better known with the German name that is Matterhorn. Is this one is very recognizable, as you can see, for the shape. And this is the third highest mountain in the Alps, and then this is not like one of the highest, but it's very important, the Monviso, because it's the source of the Po River is in this mountain, and the Po is the longest and the most important river in Italy. So this is just a video to show you, like, briefly the formation of the Alps that happened around 65 and 2.5 million years ago. So basically, the African and Eurasian plates they began to converge, and the African plate just went subduced beneath the Eurasian plate, so just went down, and as you can see here. And so this resulted in a significant volcanic and seismic activity, and this intense pressure and heat that came from this collision caused the sedimentary rocks of the ocean floor that just came up and originated this beautiful landscape. So basically, when we talk about the Alps, we are talking about like the bottom of the ocean that just came up. And I like to think that somehow I became a marine biologist for this, you know. So the geology of the Alps is very, very complex. I just will summarize everything with the 2 main important things that are the Western Alps are mainly formed with like volcanic rocks or acidic soil rich of silica. And on the other side, maybe you have heard about the Dolomites that are very famous. Here you have sedimentary rocks. So the Dolomites and this part of the Eastern Alps are mainly formed by the coral reef that came up. And so we have like this soil property, soil pH gradient, but we have also a very clear altitude gradient. And the vegetation changes a lot along this zone, this vegetation zone that we will see now like a bit more deeply. As you can see also here, we have forests until the so-called tree line. The tree line is around 2,500 meters above the sea level. It depends also on the face. In the south face slope, you can find the tree line a bit higher compared to the north face slope, where the tree line is a bit lower. What's the tree line? Is the altitude up which tree and shrubs cannot grow anymore. So, starting from the basal and hill zone, that is up to 400-600 meters, here the green shape here is the altitude that I'm talking about in each slide. So here you have you can find oaks and other Thermoxerophilus broad-leaved trees, so are mainly deciduous forest, where you can find some, sorry, there's a mistake here, Quercus, not Quercus, Quercus robur and Carpinus and other deciduous broad-leaved species. In the sub-mountain zone, that is up to 800 to 1,200 meters, we have broadleaf forests, again, such as oak, but also beech forest, this Fagus sylvatica, that is very spread tree in the Alps, and also some important tree for the economy, such as chestnut, Castanea sativa. They have this very nice fruit that is really good to eat and quite expensive. Then we move to the mountain zone till 1,800 above the sea level. Here again we found Fagus sylvatica, but also we start to find conifer at this altitude. So we have like Norway spruce, Picha abies, and also the Abies alba in drier areas of this mountain zone. Then in the subalpine, yeah, okay. In the subalpine zone, that is until the tree line, as I told you before, the conifer are dominating. So we have this kind of forests that are mainly dominated by Picha abies, Larix decidua, that we'll see again later, because this is a very important and specific plants of the Italian Alps, and Pinus cembra, and other conifers like this. And okay, and then the alpine zone, where the tree and shrub cannot grow anymore. And so here we have alpine grasslands, or mainly herbaceous vegetation, and some plants that have this specific formation, like cushion plants, that have this growth form to survive to the environment that is really harsh. And some very famous species, such as Leontopodium alpinum, and then we have also a lot of mosses and lichens. And then we have the nival zone, where the plant cover is highly discontinuous, as you can see here. Mosses and lichens prevail, and there are only very few very small plants that can resist to this environment, that are very, very specific of this environment, with a lot of really nice adaptations, I'm sorry. And one of these species that is really nice to see, I saw it last month for the first time, I was really happy, is the Ranunculus glacialis, that is this very, very nice species that grows up to 3,000 meters. So then, just to mention also the peatlands, they don't really have like an altitudinal distribution because it's mainly they are mainly linked to the water, and we can find many of them. They're very small and like pointedly located in the Alps, and we have both bogs and fens, and I mention this because they are very important for the carbon cycle, because they cover in globally. So in the whole world, they cover about 3% of the land cover, but they stock more than 30% of the global carbon stock. So they are very, very important for carbon sequestration and carbon stock. In general, we have in the Alps we have 4,500 vascular plant species. 8% of these are endemic, and as you can see, the Italian Alps are very important. They represent a lot of this biodiversity. We have more than 3,600 species and a lot of endemic, and unfortunately, we have also a lot of threatened species. So let's move to the carbon cycle. I think that maybe you already studied some about it, but we just see it again briefly. So the GPP, that is the gross primary production, is how the carbon enters into the ecosystems. You know what's the process for this? What's the ecological process that brings carbon into the ecosystem? Someone who try? What's the process? What do plants do? Yes, okay, perfect. Yeah, so the photosynthesis is the incoming process of the carbon in the ecosystems, and then we have three main outputs of this carbon that are autotrophic respiration, so the respiration of plants that represent about half of the input, and then we have 40–50% of heterotrophic respiration, that is mainly what's the output, most important output process. So photosynthesis and respiration, that is, we can call it decomposition. Heterotrophic respiration is mainly the decomposition. And then we have a small part that is just leached and that is lost by the ecosystem through the soil leaching. There are countless drivers of this carbon cycle, land use, fire, litter quality, a lot of them. And of course, in every study, it's important to take most of them into account, but it's not always possible to look at all these drivers. And I'm just put the attention on these two parameters. That are the parameters I am going to talk about. It's the GPP, as I told you, is the gross primary production, is the amount of carbon that is enters the ecosystem through photosynthesis. And then we have the NEP, or net ecosystem productivity, that is the amount of carbon that is assimilated through photosynthesis minus the respiration, both eutrophic and heterotrophic respiration. So it's basically the amount of carbon that remains in the ecosystems. Here, just one slide to show the plant carbon sink in the Alps. So before showing this value, I just want to make clearer the difference between carbon sink and carbon stock. So the carbon sink is the amount of carbon that enters the ecosystem per area per year, usually. The carbon stock is the amount of carbon that you can find in an ecosystem, in a specific ecosystem in a specific moment. So you can measure the carbon stock, but it's different from the carbon sink. The carbon sink is the capability to take carbon. The carbon stock is how much carbon we have in that ecosystem in that moment, okay? So this is the carbon sinks of the plant vegetation, alpine vegetation. As you can see, the productivity decreases with altitude because of a growth form. Here you have trees, and also for the density of the vegetation. And here, just to help you to visualize, is the one dice is the volume of 20 gram of carbon. So as you can see here, the lowest productivity is about 2 dices of carbon in the alpine zone. Sorry. Okay. Yeah, I was not going to explain that slide, so it's fine. So, which are the main problems in the Alps regarding carbon cycle that are affecting carbon sequestration by our ecosystems? Land use change and climate change, of course, that are part of the global change. And in particular, in the Alps, regarding land use change, the most important problem is that people, farmers, are just abandoning. So the livestock are not used anymore. People don't want to live anymore in the mountains because it's really harsh life. So there are no young people that want to keep going with this work, and so the land are abandoned. And as you can see, this involved the process that is called encroachment, wood encroachment. I don't know if you have ever heard it. It's when woody species recolonized abandoned areas, because you can have two types of grasslands. There are subalpine and alpine grassland. The subalpine grassland are not natural, so they are there because the land is managed. And when you abandon them, then you have the recolonization by shrubs and trees. The alpine grasslands are natural, and they're grasslands that are where the trees cannot grow. Okay, in the case of subalpine grassland, that are the majority of the alpine grassland, we have this problem. Regarding climate change, the main problem in the Alps is not temperature actually, but is drought. On the Western Alps, the drought period are creating very, very strong problem in the carbon cycle. So which are the methods that we can use to study carbon cycle? There can be very expensive or very cheap. Very expensive, of course, is the Eddy covariance method that is based on these very, very sensitive sensors that can register the amount of CO2 and water vapor that goes out and inside the ecosystems. So you have daily monitoring of all the carbon fluxes between vegetation and the atmosphere. And then you can do soil analysis to see how much carbon you have in the soil. You can use the plant trait measurements that are kind of proxy of what is going on with carbon, like biomass or specific leaf area, carbon nitrogen ratio in the leaves of your species. You can use chambers that can be fixed, installed in the field, or that you can bring around with a really heavy backpack. And you can use models, of course, and remote sensing, that is the wider method you can use to upscale your data to a regional or global level. And yeah, because yes, it's very sensitive. So the spatial scale can change. You can study the carbon cycle at the leaf level, at the individual level, or ecosystem, regional and global. And you can also use your ground data at the individual level to upscale at wider level. And there is also a temporal scale. If one of you will become an ecologist, will realize that what you think is never what you see in the field. And so to understand how climate or global change is affecting your ecosystems, you need to have very different conditions, many years of data. So you have two options. You just wait, and you hope for drought that is very bad. I feel very bad sometimes, but please, I want 1 year of drought to see what the ecosystem does. Or you can just manipulate your systems. And in this case, we use open-top chambers, as you can see here. They just passively increase the temperature inside. And as you can see, we caused an anticipation of the snow melt and so on. So first case study. I think this is the most comprehensive study that we did because we were able to put inside this study many different type of data from the individual level to the ecosystem level with different methodologies. And we work on European large forest, Larix decidua. Now I'm going to talk a little bit about this tree. And we try to answer these questions. How these forests cope with climate extremes at high elevation, and if the responses of the single tree was the same of the ecosystem response, and what was the relation between photosynthesis and tree growth during climate extremes. So we work on this, we work on here. Torino is here, here is the Piedmont region, and here you have a really, really small autonomous region in the Alps that is called Aosta Valley. And we work on this forest, this large forest. As you can see here in this picture, this species is a conifer. And do you see anything strange in this picture? If I tell you that this is a conifer? What's strange here? It's not green. Who said that? Yeah. Okay. Well, yeah, it's not green. Larix decidua is decidua, the species name, because it's the only European conifer that is not evergreen. So this is a really nice exception that makes this species very important for mitigation policies because it's very common in the Alps, and it's important for carbon sequestration because every year it grows the leaves again. So it's a lot of carbon sink, sorry. So we did ecosystem level observation with these eddy-covariance sensors, as I told you before. We look at the canopy development, so the greenness of the canopy with the NDVI, and then we did tree level observation, like the stem growth, phenological observation, and also we monitor the sap flow and the tree water deficit. The sap flow is the flow of the water inside the xylem vessels. Okay, and this is affected by drought. When you don't have water in the soil, you can see that the sap flow changes. And then we look at the meteorological data. The study period was from 2012 to 2018. So this slide is just to show you that the climate change is happening, is occurring, and these are in red, you can see the years of the study. As you can see here, the temperature was higher, and also some years, like 2017, that is the year we are going to focus on. So from this moment, you just have to keep your attention on the red curves, on the red bar plot. Was very warm, but also very dry. The vapor pressure deficit was, the anomaly of the vapor pressure deficit was very high compared to the previous years, and also the water deficit was really high. So we look also at another important thing that you have to take into account when you look at the climatic extreme is also the timing of this, when drought and heat waves happens, because it depends on the phenology of the plant. In some phases of the plant phenology, these climatic extremes can be much stronger. For example, if you have this extreme during the needle elongation, during the formation of the leaves, the climate extremes have a really more, the magnitude of the impact is much higher. So we look at this VPD and water deficit during these phenophases, and as you can see, during 2017, we have this high vapor pressure deficit. That is, vapor pressure deficit is basically how much dry is the air. The more the air is dry, the more the plant transpirates. And the water deficit was also higher during the needle elongation phase. So we expected a strong impact of these climate extremes. And also, as you can see here, you have the growth, and you can see that the growth period was much shorter compared to the other years. So we look at the stem growth. And our hypothesis was confirmed because we expected that the tree experiencing this drought would grow less. And as you can see here, you have the stem growth daily. At some point, the tree just stopped to grow. So the ring that you find in the stem is smaller. And then something was strange, because then we look at the subflow, and we thought if the tree grew less, then the subflow should have some changes, no? For example, like less subflow, or because of the drought. But as you can see here, we didn't observe anything strange in the daily subflow, so the water was keeping going through the stem. But then we look at the tree water deficit, and as you can see, the tree water deficit was very, very high during the 2017, and you see that this curve just goes down. So basically, what this plot told us, that the trees were experiencing tree water deficit, but they were keeping transpirating, but they stopped to grow. As you can see, we had almost 20% of growth less compared to previous years. So what was our expectation? That the carbon sequestration was also less, because the trees were growing less, so there was less carbon in the stems, so less GPP, less carbon input in the ecosystems. But that was not true. Actually, we found a positive anomaly, as you can see here. So in 2017, the ecosystem sequestrated more carbon compared to the average of the previous years. And we look at, with regression linear models, to the correlation between stem growth and the carbon that was sequestrated, and we found no relation at all. And this was very surprising, because most of the models that model like carbon sequestration, that use trees to model the carbon sequestration, they use the stem growth. But we didn't find any correlation between these two. What happened? Does any one of you have some hypotheses? What can a plant, how can a plant use carbon? To grow leaves, to grow the stem, but in this case, they take the same amount of carbon, but they grew less. Where did they put this carbon? Try. Yes, in the roots. Yes. They put there was a shift of the carbon allocation, because trees were experiencing tree water deficit, as we saw, so they needed to look for water, and they just put the carbon in the roots. And in this way, they just increased the length and the depth of the roots, and they were able to find water, and that's why the subflow also didn't change. They just went to look for water. Why? Because you have two different behavior in case of drought, and this is very important to understand which one of these behavior is adopted by the trees or the plants that you are studying, that you are using, because this affects a lot the carbon sequestration. So you can have isohydric plants. So the focus of this is that the water and the carbon use the same entrance or exit in the plant, that is a stomata, okay? So you can have, when you have drought, you can have two types of behavior. You have isohydric plants that when they're experiencing drought, they just close their stomata, so they reduce transpiration, so they can save water. But in this case, they photosynthesize less. And you have, like, in your system, you have like a decrease of the carbon sequestration. If you have anisohydric plants, like Larix decidua, they maintain the stomata open, and they keep transpirating, and they just try to find water somehow. Both of these strategies have two problems. If the drought is too strong or too long, in this case, the plants just risk to experience the carbon starvation. That means that at some point they are not photosynthesizing more, and they just finish their their carbon stock for their metabolism, and they die. Or in this case, anisohydric plants, you can have at some point the soil hydric potential goes so down that the air goes into the roots and in the xylem vessels. These air bubbles, this is called xylem cavitation, is when the air bubbles goes into the xylem vessels and they interrupt the water flow. The risk of this depends also on a very important plant trait that is important to measure, that is the stem density. The more you have a wide xylem vessel, the more it's easy that you experience this. It's like when you drink Coke with your straw. If the straw is big, when you are at the end of your Coke, then it's easier that you get air, okay, when you're drinking. So it's the same. The xylem cavitation is the same process. So how we are going now? What are the next steps? We would like to have to upscale this study. So we are using the LEADER system, that is the laser scanner, that, as you can see here, it gives you like a 3D model of your systems, of your ecosystems, and you can use it. It's very high resolution. You can use it to measure the stem and also the canopy shape and a lot of other parameters that you can use to you can use this data to upscale to the whole ecosystem, your observation. We also put another sensor to see to split what trees do from what the sub-canopy, so shrubs and herbs that are under trees, are doing, because we also don't know how much this sub-canopy is important for this. And as you can see here, the dark green is the sub-canopy gross primary production from 2018. We have this data, and as you can see, it's 1/3 of the GPP of the whole ecosystem. So this is important for the management of these ecosystems, because it means that an open forest that let sub-canopy grows is more efficient in carbon sequestration. If you have like a closed canopy, it commonly few plants can grow. So you don't have this buffer effect, because you can have that, like in the coffee maybe plantation, you can have that the sub-canopy respond differently from the trees. So you have this buffer. We are also looking at the carbon allocation, as I told you, at the cellular level. Here is one ring of growth, and you have many rings, as you can see here. We are looking at the cell number, at the cell lumen, and at the cell wall. Everything that is red in this picture is lignin. And the amount of lignin that you have around the cell affect the carbon sequestration of the ecosystems. And then, yes, we would like to look at the fraction of carbon that are allocated in the root system, but we have no idea of how to do that. So we are just focusing on all the other fractions, just to say, oh, okay, what remains is root. But yeah, if you will have any genial idea, please tell us. Then, how long have I been talking?
Maybe
10 minutes more? Okay. So this is much faster, and in this study we compared grassland and forest. So the forest is the same that we saw before, and the grassland is a subalpine grassland that was abandoned in 2008. So we look basically the same parameters, and okay. And so this is a species-rich Nardus grassland, and this is a priority habitat that is listed in this directive. It's a European directive called Habitat Directive that lists all the important habitat of Europe. And this is very important for many reasons. It's very, very rich of species, and some of the species, like Arnica montana, are very important for their use. Arnica is used here as well, yeah? Yeah, okay. So you know it, and then there are also a lot of wild orchids as the one that you see here. And it's very important also for pollinators, and we wanted to see if the effect of climate change on this kind of ecosystems. Again, the same data, and in this case we have also the 2022, because we have more data in this study that is not published yet. And as you can see, the 2022 was even worse than 2017. The temperature was higher, the temperature anomaly, and the soil water content, the drought, so was much stronger. So we look at the response in terms of carbon sequestration of the forest and of the grassland. And as you can see, the data of the positive anomaly is confirmed in the forest also in 2022. The forest just sequestrates more carbon. And this is nice, but it is also important to understand how long the forest can do that. So it means that this kind of drought is still like the large forest can still cope with it, but we don't know what's the threshold, because at some point this is going to change. And then we have the grassland, and as you can see, both in 2017 and 2022, the GPP was lower. So the herbs, the grassland plants, were not able to cope with this drought. This was also clear from the phenology of the ecosystems. So you can see in the same moment, during the drought, the forest was very green, and the grassland was completely yellow. So the growing season ended before, and the GPP was then affected by this. And as you can see here, we are like in August, and usually this senescent landscape, you have it like at the end of September in the Alps, so it was much earlier. And the difference between, so this is a very important parameters, that is the difference between precipitation and evapotranspiration that explain how much water is losing the ecosystem compared to the water that is entering the ecosystem. And as you can see here, the grassland has a much higher range with lower values in 2022 and 2017 compared to the forest. And we also looked at some plant traits to explain this, and what we found is that biodiversity functional biodiversity is very, very important, but we already knew that, but when you find it, it's really nice. So like in the forest, the green one, you can see that the value of this plant trait, the leaf dry matter content, if you want, we can talk about it later, is higher, but also the range. The range of value of some plant traits is very important, because it means that in these ecosystems you have more diversity, a higher diversity. And the stem-specific density, the one that I told you before about the xylem vessels, is higher in the forest. And also we found the clue of drought stress in the grassland with this trait, that is the carbon isotope. So I'll tell this because you're students, so it might be interesting. The isotope carbon-13 grows in percentage in leaves and stems, so inside the plant, when the plant is stressed by drought. Because when you have like normal condition, the enzymes that act in the photosynthesis, they prefer the carbon-12, because the carbon-12 is lighter from an atomic point of view. So it's easier for the whole process. It's easier. It takes less energy because carbon-12 is lighter. Carbon-13 is more heavy, so plants try to avoid it. But when the plant is stressed and is experiencing drought, the enzymes don't work well anymore. So inside the plant, you have an increase of carbon-13 because the enzymes are not able to discriminate anymore between these two. Last slide. Okay, last thing that is interesting is about the respiration. I don't have time, of course, talking of everything, but decomposition is very important in determining the carbon sink of an ecosystem. It's not just photosynthesis. And you can see it from here. If we focus on the net ecosystem exchange, that is the GPP minus the respiration, in 2017, we have a positive anomaly, and in 2022, we had a negative here and a positive because the NEE is minus. So when the values are with minus, it means that the carbon is storing, is a sink. The ecosystem is a sink in that case. So we had less GPP, less carbon sequestration in 2022, but the ecosystem is still a sink of carbon. It didn't lose because the decomposition was lower, because the drought impact the composition as well. So if there is a threshold of drought that is like depends on the ecosystem, after which the microbes in the soil are not able to decompose anymore or are much slower. So yes, we had less carbon sequestration, but we had also like a decrease of decomposition rate. That's why in this case the ecosystem was still acting as a sink. I'll skip the third study. And it just, I made like a really huge like landscape of a lot of things that we can take into account. And this study was just about how, because I don't show everything, but I just say there's like a big discussion on alpine among alpine ecologists. That is it better to have subalpine grasslands that are managed, or is it better if the forest go back and just take again all the because we see that the forest is much stable, no? So why should we take this kind of habitats? And we did this study, and that's true. The invasion of shrubs are making the grassland more stable, but there are other problems. That is that we're losing species, that the forest transpirate more, so you have less water that goes in the soil, and that water supply for humans, for example, and pollinators, and also another important problem of shrub encroachment. That is in the Alps, not very much, but in the Pyrenees, for example, this is a project that I did in the Pyrenees on shrub encroachment. It's it's a huge problem for fire, because grassland are like this. When you have like a mixed landscape, the fire it's hardest for the fire to go through the landscape. But if you have only shrubs or forests, then it's a very huge problem, and they have this problem with this species that is encroaching their subalpine grasslands. It's a legume species with spines. It's very bad. As you can see here, this is the situation, and they are coping with, and that's it. Thank you. I think I'm gonna stop. So thank you for listening, and I'm really happy to collaborate with everyone. And so here you have my email, and write me for whatever. Thank you. Hi.
Thank you for your presentation. Very nice. I have a question about the soil composition and the soil take samples. For example, what depth you take the samples to make the measurements
Of the soil, you mean?
in the soil in the lab?
Okay.
The first 30 centimeters, or you make a complete soil profile to measure the carbon sink or the carbon stock?
Both. So we have a pedologist who did a soil profile to characterize the soil, and then we took soil core that were 15 centimeters deep and 5 centimeters of diameter. We took many of these soil, like randomly located in the systems, and then we, in the lab, we split it in organic horizon and A horizon, A soil horizon, and we did the analysis on both organic and A horizon.
But only in the 15?
Yeah, yeah.
And why don't you make a complete profile?
Yeah, because I think that this is linked to the system. That is very different from your kind of ecosystem. I guess you have like much more soil in your like tropical or this kind. In the Alps, after 15 centimeters, you don't find really a lot. Yeah, sometimes you have like, you can have also rocks. Yeah.
The next question is about the soil respiration. When you use the chambers,
Yes.
I want to know the time that you that you put the chambers.
Half an hour.
Half hour.
Half an hour.
Every
Maybe
day.
more.
Every day. Along the year.
No. no. Half an hour once a month per month.
Once a
Per
month.
month, yes. And we try to do the 1 week of sampling per month, and with more measure for the same plots in different times of the day. So, like for example, we started from plot A to plot zeta, and then the day after we did from the zeta to the A. Yes. Yeah. It's the one that is the one that moves. Yeah, with the backpack. And then we had also the one that fixed in the field, but we used that one only to compare with the eddy covariance. They were like a kind of tarare, non so come. It was to see if the sensor of the eddy covariance were working well, like to have ground truth.
Yes,
Yeah.
the eddy covariance is still in the same place.
Yes. Yes, that's fixed and is measuring. Yeah, is measuring daily. Actually, every 30 minutes. Yeah, every 30 minutes. Yeah.
Okay, and the last one.
Yes. To calibrate. Yeah, to calibrate. Yeah. Thank you.
Okay, and the last one, when you show us the results of the carbon sequestration by the forest, maybe, I don't know, maybe it's a suggestion,
Yeah.
maybe the sequestration by the forest, by the climax forest,
Mhm.
is not the trees, the mature forest cannot sequestration more carbon. Maybe the youngers, or maybe the
You mean the subcanopy?
yes.
Yeah,
is the-
that's
is
why you
that,
put the-
that's
Mhm.
your answer.
Mhm.
That's why you have that sequestration in the
Yeah.
forest, because the mature forest cannot sequestration more.
Yeah. You mean like that the subcanopy has had like
Yes.
a buffer effect during drought, or it might be that's why you put the eddy covariance also in the subcanopy to try to split. The problem of this is that you cannot split the respiration, because you can see how much GPP goes to the tree, because we put one sensor above 20 meters on the canopy, and then one above 2 meters, so you look only the subcanopy, but the soil is the same. So you cannot divide you can see the GPP, but you don't know who is respirating more, if trees or shrubs and the herbs. Yeah, that's the challenge.
Thank you so much.
Yeah, thank you. If someone is shy, you can try to ask in Spanish. I understand a little bit, maybe.
Hello.
Hello.
When I did see you and hear you presentation, I thinking about the mycorrhizal fungi, and my question is follow: what kind of consequence you can thinking about the dynamic about the mycorrhizal dynamic. With your datas of the ecosystemic measures, in the case of the coniferous forest.
Okay, you mean how the symbiosis changes during climate extremes?
Yes.
For example, I
Yes.
have no idea. Yeah, I mean, there are some studies demonstrating that these symbiosis increase during when plants are stressed, but we didn't study it, and it's something that we really would like to do. But you know, also if you do that in the field, it's very, very complicated. We have some soil samples that we are going to use to describe the biodiversity in the forest, but how these symbiosis respond to climate extremes, no idea.
Okay, thank you.
But you're welcome if you want to come to
Thank you.
study. Yeah. Hi.
Chambers that you use to simulate scenarios of global warming, how much increase the temperature?
How?
How much increase the temperature?
I think it was, let me think. It was like 2.5 degree. Yeah, not more. It wasn't very much, but enough to see something in that ecosystem. Yeah.
So it's a lower scenario of
Yes.
global warming.
Yeah,
Okay.
yeah.
And how long should be carry on the experiment?
Until
whole year?
they got destroyed by snow, the open top chambers. Yeah, we put it. It was like 2 years to 3 years. 3 years. Yeah, and we were using it to study the decomposition. So we put then litter bags inside the open top chambers and outside, and we look at the decomposition to see if the drought if this increase in temperature would increase the decomposition rate, and we actually found that the decomposition was less because the soil was drier with the soil temperature. So it's evident that you cannot really divide temperature and water. They're very important altogether. And in that experiment that is not published yet, we also found that if you have, so inside the litter bags, we put different type of litter, just one kind of litter and more plants, more different plant litter together. And we found the proof of synergic effect. I try to explain clearly. Like you have species A, B, and C alone, and we put A, B, C together. The decomposition of the litter of A, B, C together was higher compared to the sum of A, B, and C alone. So it means that when you have like a mixed litter, you have more ecological niche for microbes, and they act differently and when they are together. It's not the same if you study the plant A, B, and C, and then you just sum them. It's different. Yeah. So we found that this biodiversity was like responding better to the manipulation. So again, the diversity is important.
And usually the models of the threshold models use water potential and temperature to simulate the changes in some parameters like germination. In alpine vegetation, have you tried water potential too?
Yes, we are working on it, but we didn't have the result about the modeling already. We are trying to do it with the Aosta Valley. They want to do that at regional level. So yeah, it's easy with them because the region is very small and it's an autonomous region, and it means they're richer and they have a lot of more like data and instruments and everything. So they're trying to do this modeling at the regional level, but I don't have anything yet. Yeah, thank you.
¿Alguna otra pregunta? María.
Thank you. So if you were asked to do a similar study, but using the Mexican drylands as a model of study,
The Mexican?
the Mexican drylands.
Okay.
So what would you change in the methods, or how would you use your experience in the Alps to improve some methodology here?
Okay. And if we consider that, should I consider that we can have all the methodologies or just the like, because the best
in a perfect world.
Yeah, the best is always to have the sensors. Yeah, that's, and you can, the eddy covariance is for everywhere. It doesn't have any limits. So the best thing is to use at least chambers and go there and do some measurements. And yeah, then of course you can use also some proxies as plant traits, but you need data to correlate plant traits with. So you first need to measure. It's yeah, you need to measure, or otherwise you can just cut. You can just cut and put the biomass in the oven and then measure how much carbon you have and try to do it with the most important species that you have. And usually the rule is that you use the most dominant species up to 80% of the cover of the ecosystem. So you pick the species that sum together give you the 80% of the cover of the ecosystem, and you look at the traits of that. So you can pick the species and cut them and see that's a beginning. But if you want to be very, very sure of what you are measuring, is the sensors. Yeah.
Okay, thank you.
Yes, thank you. How many times do you make the calibration of the sensors?
Once a year.
Once a year.
Once a year, but I don't do that. There's a person that is this wonderful woman. She's specialized in eddy covariance, and she does the calibration. Yeah.
The last one, you can measure, or you measure another greenhouse gases besides. Do you measure other gases of greenhouses?
Greenhouse.
Besides CO2, for example, methane.
It measures also methane as well, and I don't know if some nitrogen something, but yeah, I didn't use those data, but yeah, they can measure other gas, yes, for sure. Mm-hmm.
Thank you so much.
Yeah, because they're used also to see like the impact of like grazing. So you have also other gas important for the greenhouse. Yeah, other greenhouse gases. Yeah. Okay. So thanks to all of you for being awake.
Okay. Si no hay más preguntas, a ver.
Oh yes. Nice. Yeah, I'm happy.
Thank you for your presentation. And final one, regarding all this data in, let's say, a multi-research group in Europe.
Yeah.
All this data, where are they leading to? Is there some kind of mitigation actions that are actually taking place right now because of all the data that you are achieving? Is there some kind of policy that, okay, then what's the next step? You know, because what you said, we don't know what's what the ecosystem is going to do next. You know, which will be the behavior of the ecosystem in the next years, which the drought is beginning to be harsher, and let's say at a continental level in the union, is there some kind of policy, or have they been you have already in place in some places of Europe some other, let's say, next step actions with this data?
Yeah, so I don't know if I can really answer to your question, because I work mainly at the regional and national level, so I don't know. But of course there's a network, and there is also, we did the European meeting in Rome last year. And that is all these data are in the network that is called FluxNet, and there is the ECOS, that is like the scientific organ, yeah, like the scientific committee that works with I forgot the ICOS, yes, ICOS, that is the scientific committee that works with this data, that meets every year, and also with people representing the political part. Okay. So we meet, but then how every states use this data, or Europe, I don't know. I just know it at the like regional and national level.
But you have an experience as a group as a research group. You have an experience somewhere, some kind of intervention of the area, of a specific area, based on the data.
Sure. Yeah, okay. Yeah. In the Aosta valleys, they are like managing pastures and forests according to this report of this data.
Okay.
Yeah.
Okay. Mm-hmm. Thank you.
Yeah, it happens sometimes.
Yes. Okay.
Yeah.
Okay, muchísimas gracias. In the streaming we have any questions. We just have greetings and congratulations for you.
Okay, thank you.
So, le agradecemos a la doctora por su interesante ponencia y pues esperemos continuar con esta colaboración. Muchas gracias. Un aplauso.
Gracias.